Welcome. I hope you really enjoy this day. It is not meant to be stressful at any moment, but it is very good if you can contribute and ask questions of the speakers and really bring it to life. Show us how excited you are by what you're listening to, and it's a once-in-a-lifetime opportunity to ask some questions. So this brings me to my first plenary lecturer, which is Professor Mark Dawson, and he's a physician, scientist and program head and Associate Director of Research at Peter MacCallum Medical Cancer Centre. And he honestly is, I think, the most active publisher in health and medical sciences in Australia today. He'll deny that, but that's my observation. It's like a paper, I'm probably undercalling this, every few months in Science or Nature. The rest of us just dream of that, just to tell you. Mark's work is studying chromatin regulation in hematopoiesis and cancer, and he's defined key molecular mechanisms that underpin initiation, maintenance and progression of cancer. And this has led to several first-in-class epigenetic therapies that have translated into trials around the world. And he's got an investigator grant, but Mark is so much more than an investigator grant. So I'm very excited that I'm actually have the opportunity to have him here today. And Leila, I'm not getting your connection. Can you come and save me? Where's she gone? Oh, yeah, I'm not getting the little hand. Oh, no, gone backwards. Here. Oh, now. Thank you. Mark, please join me.
Thank you so much, Ingrid, for that very generous introduction. And thank you also for persisting with me. Ingrid has invited me, gratefully many times to come, and unfortunately several times I've not been able to attend, but today I really wanted to make it here. And it is really a testament to both her passion and vision that we have this forum. I would have loved to have been in the audience like you are 25 years ago to meet people who I could have pinned my mast to, I think, in many ways, and said, that's the kind of career I would like. So today I'm going to tell you what drives, I guess, me, and how did I get to, this current position in time. And I really want to focus and emphasize the importance of scientific curiosity in this journey. The term physician-scientist, in many ways, has a very broad definition, right? But I want to tell you how I would accurately describe myself. I would describe myself as a scientist that is clinically active, okay? Because I think what I feel like my purpose on earth is for discovery science. And what clinical practice does is provide me with a vision of what the key questions are to go and answer. And so when you think about how many people around the world do a job whereby they're coupling clinical practice with running a discovery science laboratory, which is the key engine for their discoveries. Actually, this article in the New England Journal of Medicine, I'd really encourage you to go and read. The answer is, if you look at all of the university graduates that will leave the University of Melbourne, Monash University, any other university around the world with a medical degree, less than 1% now, this is a few years old, but less than 1% of them will actually go on to try and couple a career where they are a scientist, but also seeing patients. Why is it important to really preserve this field? Well, if you look over history, it says that although there's less than 1% of us that do this, more than 40% now of all the Nobel Prizes in physiology and medicine have been awarded to this group of people, because they come with a perspective of what the key problems in medicine are and use their scientific training to try and solve that. However, as I've tried to allude to, this is really declining, and there's a lot of reasons for why this is declining that we'll get to. And I'd really encourage you, if this fire burns in you, to really take this on. This is not a linear path, and I wanted to go through the road I travelled to kind of get here. So I finished high school in 1992, so that dates me. And I started across the road at the University of Melbourne at the age of 17. I had no aspirations of doing medicine when I was at high school. I did far better than I expected to in my year 12, and on the guidance of many of my teachers and my parents, neither of whom are medical, but felt that I was a really good communicator, thought that I would be able to contribute in a way by doing medicine. Right? And so I enrolled in this degree, and the first 3 years, I appreciate the degree has really changed, particularly at Melbourne. It preserves some of its origins at Monash. But in the first 3 years of the preclinical degree, I just loved it. You know, I loved learning everything there was about the human body, physiology, biochemistry. Those were, you know, really formative years. But then I went on to do clinical school. And there it became really apparent to me that I potentially had chosen the wrong career. Right? Because my life had always been about black and white. Right? Yet medicine is very much grey. Right? And what do I say? What do I mean by that? You know, symptoms such as headache, chest pain, abdominal pain, etc. There are many, many reasons, and probably the least common reason for chest pain is angina or pulmonary embolus, etc., etc. Right? And it's very difficult to get to the root of this problem unless you're a great communicator. And probably at that stage, I felt that I was too young to get the best out of myself in that realm. So I made a decision after my 5th year of medical school. I remember clearly the day I just finished my obstetrics training and thought, this is not for me, right? And decided to leave, and I actually went to the University of Oxford to do science. And had committed that this was actually, you know, got a scholarship, went there, and this was going to be my future. But for a number of other reasons, it did not work out at the University of Oxford. And here I learnt what it was like, for the first time in my life, to categorically fail at something. And so ended up back in Melbourne, not a medical student, not a scientist, aimlessly wondering what is next. Right? And then I met this man, who was introduced to me by a colleague of mine at med school. He was at the Walter and Eliza Hall, where he actually did some of his formative training with Don Metcalf, and was the postdoc involved in what became one of Australia's greatest contributions to the medical field, which was the discovery of cytokines such as GCSF that we use every day in cancer medicine. Okay? His name is Bill Robinson. I remember clearly he introduced himself to me as a physician-scientist. I'd never heard that word before, you know, and I wondered, I actually nearly asked him out loud, what kind of job is that? He was here on sabbatical and needed people in the lab. And he was just an incredible personality, you know, one of the most charismatic and endearing people you would meet. So he took me on. He never asked me ever why I came back from Oxford, right? He knew that was a pretty traumatic time in my life, but I guess his way of not asking me about it was his way of showing me that the decision was inconsequential. And what he did was, we would do science during the day. We worked in a model organism called zebrafish. At the time, understanding the cell cycle was exploding, and we were working on regulators of the cell cycle in zebrafish, which go from a single cell to a swimming vertebrate essentially in a day, right? So you get to observe how the cell cycle progresses in great detail. And every week he would take me to his clinic just to sit down and hear him talk to patients. And within a year, he'd mended all of the things that were broken, right? And he had convinced me to go back to doing medicine, right? Which I did. And I went back to St Vincent's Hospital to finish my training. And then went on to an internship and residency, I still struggled with the grey, right? And it is in part that, and it is in part Bill's inspiration that helped me choose my career, which is as a haematologist. Because in haematology, nearly everything can be measured, right? And everything can be explained, and you do a dual training, right? You train as a pathologist and as a physician. So I did my training at the Alfred Hospital and trained there as a haematologist. And this was just a fantastic time in my career because, you know, I was able to couple training in pathology, seeing, diagnosing the diseases, but also the clinical management of this. Never once did that curiosity fade. I always knew that I couldn't just practice medicine. Right? I had to do something else. And fortunately, along the way, I met my lifetime partner, who is also a physician-scientist. And together with her, in 2005, we both got independent scholarships. And this time for me to return to the UK. But this time, I chose the other prominent university there, at the University of Cambridge. Right? And this time I went to work with a physician-scientist himself, Tony Green. So Tony was a haematologist who did his postdoc at the WEHI under the tutelage broadly of Don Metcalf and Glenn Begley. Right? And Tony was fantastic, and he got me to work in a number of different model organisms asking discovery science. What he asked me was, what do you want to learn out of here? And my answer was, I want to know whether I'm good enough to couple this career. Right? I don't want to do a correlative clinical medical project because I could have done that in Australia. Right? I want here to test myself in an area that I'm totally unfamiliar with. So he recognized that I'd previously worked in zebrafish, thought I might be useful in Drosophila, a completely different organism, and got me to go and make model organisms, Drosophila, of what his lab had helped discover at that time, which was that a gain-of-function mutation in an enzyme called JAK kinase led and underpinned, 99% of all of the myeloproliferative diseases that present as polycythemia vera, which is too many red blood cells. For 50 years, we had known that this disease was related to other diseases, but the molecular pathogenesis of this had not known until 2005, when his lab and 3 others made this discovery. And so I started working there when a paper was published in a journal called Nature Genetics in flies, saying that this kinase alters the structure and function of chromatin. And that led me to meet this man, Sir Tony Kouzarides. So Tony, at that time, ran a laboratory working on chromatin biology, and I'll get into what chromatin biology is. And I'd say about 80% of his lab worked in yeast, where many of the major discoveries had been made about how chromatin is actually regulated. I was one of the first physicians to walk into his lab. I was the only medically qualified person in the whole institute that they worked in. But what it allowed me to do was just immerse myself wholly in discovery science. And with the support of the Wellcome Trust, we made a number of major discoveries that really set up what I wanted to do for my enduring career. And then there came a decision about where to do this. I still felt very strongly about Australia and Australian science. And Sarah and I decided in 2014 to move back here with our 2 kids, predominantly because of the promise that this building offered. Right? What might not be necessarily apparent to you is just how special this precinct is in terms of its critical mass. Right? You have the University of Melbourne, the Florey where we're standing in, the Walter and Eliza Hall, the Children's, the Women's, the Royal Melbourne Hospital, the Peter MacCallum Cancer Centre. You know, there is a concentration of intellect here that is really unparalleled, I would say unashamedly, in Australia, and comparable to many of the great places in the world. And this is why I think this precinct, you know, holds the promise for many great things to come. So before I came, I decided what Bill made me realise was that I would make a commitment internally to myself that there will always be a physician training in my lab, and mostly this would be someone who was unclear whether clinical practice was the only thing they wanted to do. The first of that, and this was who we moved back with, his name is Chun Fong. He was a haematologist who was training in Melbourne, who came to the University of Cambridge to start his PhD with me. I'm incredibly proud of Chun because he, you know, not only smashed his PhD out of the park, but he is now the Director of Research for Haematology at the Austin Hospital. And Chun really helped set the culture for what is now our lab, right? And so I run this now very closely with Sarah Jane, who is in the middle of this picture here. And together, you know, we train a whole number of people across various fields. But let's get into what my field is, right? I wouldn't mind if some way that this was relatively interactive. So my field is the field of epigenetics, and whenever I give a talk to an audience like this, I show this picture, right? Because this, in a nutshell, summarizes what our work is. Everyone here knows that the caterpillar becomes the butterfly. But what you might not necessarily take the time to appreciate is that the DNA content is exactly the same. Every single base pair that the caterpillar has is the same DNA content that the butterfly has, right? Yet, you know, a child can often think of this as two completely separate species, organisms, etc., etc. And this difference in outcome, not only phenotypic, behavioral, etc., is driven by access to that DNA template to express the genes that are required at the right place at the right time, to repair those genes and to replicate that DNA content. So access to that DNA template, by and large, is the study of epigenetics. So let's drill into this and give you some more facts, right? If you were to stretch out the DNA content of any one of you sitting here, in fact, any human being, right, you could walk to the sun and back 600 times, okay? So consider that for a moment and consider the problem that we now need to solve, right? Which is, how do you find places of damage and repair it, right? How do you replicate this absolutely faithfully? And how do you get to the right place at the right time to express the right genes? Right? The DNA content of your blood cells are no different to the DNA content of your liver cells or your skin cells, right? And all of this is driven by this process of access to the template. And by and large, we can drill down and say the way DNA is packaged in our cells is largely in two states, right? There is the more open conformation of chromatin, which we call euchromatin, and this is where active genes for that particular tissue reside, right? So in blood cells, they will be the genes that give that cell that identity, right? And the genes that give a liver its identity will actually be shut off, not lost, but shut off, right? And that is more closely packaged as heterochromatin. Now, this one genome, different phenotype, is really what describes what my lab does, right? And we use, in many ways, the hematopoietic system to study this, because the hematopoietic stem cell, which sits at the hierarchy, actually drives the genesis of hundreds of different types of cells. I presume someone will tell me if I'm running out of time. Okay. I will do something special for whoever can tell me what is wrong in this diagram, right? This is hematopoiesis 101 as presented in any textbook, right? Sitting at its apex is a hematopoietic stem cell, and down the bottom are all your effector cells, including red blood cells, platelets, and various immune cells. Anyone want to hazard a guess as to what is wrong with this diagram? It's the arrows, okay? Because the arrows imply that cell fate is unidirectional, right? That once a cell makes a decision to become X, that is set in stone. And that's largely how this is still taught today, right? But the reality is, we have known for probably close to 70 years that this is not true, right? That cell fate is not unidirectional, right? Sure, normal development takes a well-trodden path, but that can be reprogrammed, and there is this concept of transdifferentiation as well. These are key issues that we see in cancer biology, right? Where cancer cells are some of the most plastic things that we encounter and how they adapt. Now, this concept of cell fate not being a one-way street led to the Nobel Prize for Shinya Yamanaka and John Gurdon. Who here has heard of Dolly the sheep? Right. Keep your hand up if you've heard of John Gurdon. Okay. Ian Wilmot did Dolly the sheep and is credited for it, but he did not get the Nobel Prize. John did. Because John did arguably the most important epigenetic experiment that will ever be done, right? And Dolly the sheep was just this experiment over again. So let me take you to that important experiment. John, as it turns out, was my PhD examiner, in Cambridge. And, just one of the most inspirational people you will ever meet. Right? So this is John's experiment in the late 50s, right? Around that time, Watson and Crick had just described, took the credit for describing the structure of DNA. What the prevailing hypothesis at that time was that as cells differentiated, they lost the pieces of DNA that they no longer needed. Right? So for instance, if a cell decided to become a blood cell, it no longer needed the DNA content that would make it a skin cell or a liver cell or a nerve, etc., etc. John was uncomfortable with this hypothesis and wanted to disprove it. Right? How do you disprove something like this? This is the experiment he designed. Right? So here, he took the nucleus of a fully differentiated cell, in this case, a gut cell, an intestinal cell from the frog. He irradiated the nucleus of the frog oocyte, so destroying the DNA content of that, and transferred the nucleus from the intestinal cell into the frog oocyte, wondering what would happen. What happened was, rather inefficiently, but unquestionably, what happened was that oocyte provided the right environment to fully reprogram the DNA content, leading to a tadpole and subsequently a frog. Right? This is called cloning, right? And this is how Dolly was made. Right? Dolly is the first example of a vertebrate being cloned, of a mammal being cloned, but this, in frogs, was done many, many, many years ago. Right? What this experiment unequivocally proved was we don't lose the DNA content as cells differentiate. Right? It's shut off, but it's absolutely preserved. And so it is these kind of questions that my lab aims to describe. Right? And so what I'm going to do now is just give you a brief overview of the type of work we do, and then end on a short story of something you can't read, which we're really excited about now. Right? So my lab really uses CRISPR technology which has just changed the field. And if you think of the origins of CRISPR, that was driven by curiosity, right? What is the bacterial immune defense mechanism largely drove the discovery of CRISPR, right? Yet we use it every day now in agriculture, in plant biology, and actually in medicine and discovery science, right? And so it's the main engine for my lab in terms of making unbiased discoveries. One of the other areas that we're really interested in is this concept of clonality. Every one of you, because you're now over, I can't see anyone under 20 in the room, all of your tissues have started to become clonal. We know that clonality gets more and more prominent as we age, and if we take the blood system as an example, we produce 200 billion blood cells a day. You're born with around about 100,000 to 200,000 blood stem cells, but by the time you get to the age of 80, nearly all of the people who are octogenarians are producing that demand of 200 billion blood cells a day from 1 or 2 stem cells, okay, that have acquired mutations in epigenetic regulators that give them that capacity to be able to produce that demand, right? And that's just not true for blood, it's true for skin, it's true for your lung, it's true for your pancreas, it's true for nearly all of your tissues. And understanding why clonality gets more and more prominent as we age is one of the questions we are interested in. We want to understand how cells adapt to various challenges without mutating their genome. And we have built lots and lots of tools for this because this is one of the key problems in cancer biology. The minute you administer a therapy to the cancer immediately starts to adapt. The way it adapts most commonly is not by developing new mutations. Okay, that just takes too long. It needs to adapt far more quickly, and many of that is by rewiring its genomic content. We work very closely with Sarah Jane's lab to understand intratumor heterogeneity. One of the other major areas of interest in my lab is really looking at how we can leverage the immune system to better control cancer. And finally, one of the other areas that we're really interested in is chemical biology. Right? I've highlighted here in yellow all the physicians that contributed to these discoveries. Right? So every one of the discoveries that are on this list, highlighted in yellow, was a physician who came to my lab to train to become a physician-scientist. Okay, in the last sort of 5 minutes, what I want to do is just tell you about a discovery that is not yet published. Okay? And it stems from this, right? So the most successful cellular therapy that has been around for close to 70 years is that of hematopoietic stem cell transplantation. About 150,000 people around the world every year get a blood stem cell transplant to treat various malignant and non-malignant blood diseases. Right? But that only represents about 40% of patients who could actually benefit from one. Right? And the reason those 40%, the rest of them miss out, is because we can't get enough stem cells or we can't find a donor. So the key problem we wanted to solve was how do we do this? Right? The other major problem in hematopoietic stem cell transplants is that the most common cause of death in the first 4 months post a transplant is not your disease coming back, it's not graft versus host disease, it's infection. Okay? And the problem with this is that whilst we have developed ways in which we can encourage the reconstitution of our innate immune cells like neutrophils with GCSF, T cells actually take much, much longer, and they underpin the problem with opportunistic infections. So the type of experiments my lab does is to take a blood stem cell and put in a unique genetic barcode. We then allow that cell to replicate itself many times over, and then we understand the properties of that single clone, right? and ask what are the transcriptional programs of that clone, what does its chromatin landscape look like, what is the genome of that clone, etc. And then we transplant that into multiple different mice. Now, instead of there being only one clone, imagine there's 5,000 of them, right? So then we can serially trace over the lifetime of this animal the downstream consequences of each clone. With this being the question, right? So if this red clone gives rise to B cells in all of the mice that it gets transplanted, given how long it takes to culture them and then put them into 3 different individuals, this is not by chance, right? There must be something cell intrinsic to that clone that drove that fate, right? If it gave rise to different types of cells in 3 different mice, that's being influenced by the microenvironment. So we've done that experiment many, many times over to really start to look at what it is about the environment and the cell intrinsic nature that drives the various decisions that a blood stem cell makes once we transplant it into an individual, right? So we take these blood stem cells, transplant them into various individuals, and track them. The first and most incredible finding was that if we follow this over 5 weeks, and here what you'll see is right up here on this side is, as you can see up here, this blue clone is the one that's most likely to divide in the dish. It makes up about 4% of all of the clones in the dish. But now we transplant this into several different mice. And what you will note is that this pink clone here is what we would call a multipotent hematopoietic stem cell. It gives rise to itself, it gives rise to B cells, T cells, red blood cells, myeloid cells. Right? But it does so in every single mouse. The size of that bubble tells you how much of the overall hematopoiesis it contributes to. If you haven't already noticed, this is it's almost identical in every single mouse. Right? So this can't be a fluke, this is a cell intrinsic property. This is not just true for multipotent hematopoietic stem cells. Here's lymphoid skewed, myeloid skewed, and cells that only give rise to T cells and red blood cells. So this means we've now got a system where we can actually find out the properties of cells that drive certain cell fate decisions, right? But what about T cells? For a blood stem cell to become a T cell, it needs to do something special that the other blood cells didn't do, right? Which is it needs to get to the bone marrow and then subsequently get to the thymus. And only in the thymus does it differentiate. So one of the first things we did was we took these cells and transplanted them both into the thymus and the vein. And when you transplant them into a vein, you give rise to all types of blood cells: red blood cells, white blood cells, T cells, B cells. But if you put them into the thymus rewrites all of these cell intrinsic programs and only makes them T cells. One of the postdocs did an incredibly interesting experiment where she asked, what if we put it into a mouse that's never made T cells but has the structure there for T cells, an immunodeficient mouse? Remarkably, when you put blood cells in there, it regrows the thymus, and that mouse is now able to put out T cells. So using a variety of different methods, we were able to describe several types of blood stem cells. Those that, regardless of where you put them in, either directly into the vein, directly into the thymus, or directly into the femur, give you T cells. These are called group 1 clones, these green ones. Right? Those that only give rise to myeloid cells and B cells, but not T cells, but if you put them in the thymus, you get T cells. Those that only give rise to B cells, but if you put them in the thymus, you get T cells. And those that are poor stem cells, but if you put them into the thymus, you get T cells. And now you can go and dig and find out what is underpinning this, right? And I'll get through this really quickly, but this is the gene expression program of what defines those group 1 cells. And at the top of that list is this gene called XLR4B. Don't worry if you've not heard of XLR4B. Until about 9 months ago, I had not heard of XLR4B. But I did what any other scientist would do, right? I went into PubMed and I said, what do we know about XLR4B and T cells? And the answer was this. You know, So this is an incredibly exciting time in a scientist's life, right? You found something that nobody else in the world knows, right? So what do we know about XLR4B? So if you dig in the literature, it's preserved all the way in vertebrates. It arises for the first time in jawed fish, right? There's only been really one major publication on this, and it was published in Nature in 40 years ago, where it had its origin because here, Mark Davis's group was cloning areas of the genome that were lost in mice that did not have adaptive immunity, right? And he found a set of genes on the X chromosome that he called X-linked lymphocyte related, hence the term XLR. So we thought we might have found a way to make T cells on demand. To do that, we put XLR4B into blood stem cells, put them into mice, and what do you know? It makes lots and lots of T cells. Right? So this is an example of this, right? And if you take nothing else away from today's symposium, what I want you to take away is that without curiosity, there are no discoveries, right? And without discoveries, there are no cures. This is a particularly important message at this time because our state and federal governments fail to fund fundamental discovery science, which will drive what are the innovations of tomorrow. Right? So none of this is possible without the group and the funding agencies, and you know, it's been a real pleasure to be able to talk to you today. Hopefully there is some time for some questions.
Well, thank you, Mark. That was worth the wait. How many years? 3 years, maybe more? I don't know. I've stopped counting. I wasn't going to let you go, though. I'm sorry about that. But thank you very much. I think Mark really epitomises a clinician-scientist with the emphasis on discovery scientist. But he couldn't understand how important what he's doing is without the clinician bit that helps drive your motivation and gives you special insights. Now, we have time for a couple of questions. So if there's anybody with a question.
Hi Mark, I'm Nella, I'm from Monash Health. You touched on CRISPR, but I know that the time was limited and you couldn't go through details, but could you just a little bit very quickly explain how CRISPR incorporates to your research and how you use it in your research? Thank you.
So think about CRISPR as the ability to change very specific parts of our genome identified by the barcode that is our DNA template. Right? So you can take any machinery to any place in our genome to either cut or change. Right? What you can do is knock out genes to ask what they do. You can overexpress genes to ask what they do. And now, more recently, you can start to do what is called base editing. Change the sequence of that genome. That will have, you know, incredible value for a variety of diseases that are driven by single point mutations. Sickle cell disease being a great example of that, right? Imagine you can just correct that in the blood stem cell back to its normal counterpart and deliver that cell back to the patient. You're giving them their own cell back, but now rectifying the mutation that caused so much harm. Right? So that's one therapeutic way of looking at it. What we do is we've asked other questions with that. We've asked, what if we can change the codons of every single codon in a protein to either increase or decrease its function, right? And we've chosen proteins that have really important activity in human health and disease. So I think the opportunities with CRISPR is really unlimited.
Great. Well, if there are no more, there's one more question at the back there. We'll have that one, then we should move on.
Hi, Mark. This is Purva from ONGCRI. My question is, what advice would you give when you want to select a topic or area of research? Would you advise to stick to what you already know and maybe go into clinical translational research, or as a scientist, you should explore more basic research techniques? Yeah,
it's a really good question. It largely depends on your personality. I'll tell you what I tell my children, right? Which is, if you only do the things you're good at, how will you become better? Right? So if you don't actually train yourself to be good outside your comfort zone, how will you broaden your horizons, right? But it requires you to be brave enough to do that, right? So I would really encourage you to step outside that comfort zone. By the time you've finished your training as a doctor, you're a pretty good doctor, right? And you'll become better as you train. No one's the finished product. I still learn things on the ward today, right? And you've just got to be comfortable with the fact that you're going to have that continuing education. But if you really want to kind of broaden your horizons as a researcher, step outside that comfort zone, is my advice.
Please join me in thanking Mark for a wonderful talk.